All right. Thank you, everyone, for coming to my psychology experiment today. I assume that you all signed waivers before you came in, and if not, well, your presence is signing a waiver. Today, I will be experimenting. Never mind. The title of this talk is multimodal neuroimaging a thing? Is it useful? Which is kind of a comical thing because a couple of years ago, we founded this Center for Multimodal Neuroimaging that's run out of the fMRI facility, which basically says, yes, it's already useful. So it's kind of a justification for my existence, or possibly our existence. But then again, what isn't a justification for our existence? So is multimodal integration useful? And I'm just going to go with yes. If you're satisfied with that, the doors are there. There might, well, I don't have candy, but I'd reward you with something else at the end if I could. I'm going to go over a couple of things. What is multimodal neuroimaging, for those of you who haven't heard me talk about it at least once. Some of the motivations. Then I'm going to talk about why multimodal neuroimaging is hard, and even though it's hard, why we should be doing it, and why we should be spending more efforts on making this a thing. If you're intrigued by the topics today, the Center for Multimodal Neuroimaging is hosting a workshop at the end of October, the 30th, 31st, and November 1st. And we are bringing in 15 extramural speakers to talk about a range of topics that will be included. Some of them are included in this talk, or some of the thought questions in this talk are inspired by that workshop. I highly recommend that you sign up once we get the page launched, and I highly recommend that you attend or WebEx or watch the recordings later on. So briefly, and sorry for the raspy voice, it's summer cold season. Multimodal neuroimaging is just putting together any two types of neuroimaging, sometimes more than two types. And so it might be fMRI and EEG, it might be MEG and EEG. Some people consider fMRI and structural MRI. I will not stand there. fMRI and DTI, fMRI and MEG. And then there's also these neuromodulation techniques like TMS, TDCS, TACS for doing neuromodulation that can also be paired with different imaging techniques. So you can be asking the question of what happens if I disrupt the brain, modulate the brain, enhance the brain, or inhibit the brain, and how does that change the networks or the functional things? I feel like I'm riding a roller coaster and my head's not really turning. So if we don't make the requirement that multimodal imaging is simultaneous, you can see that there is a vast number of imaging techniques that you can use. And here what I've done is sort of highlight everything in purple is sort of things that we're working on. This is really scary. I'll stand back here. How about now? Okay.
Step back where you were.
Yeah, I'm good. Yeah. So if it's not simultaneous, then we'll just say anything that is in green is possible. Things in blue are fairly well studied. So fMRI and EEG is fairly well studied, even though we're making advances all the time. And things that are in purple, we're starting to see a vast increase in publications over the last 3 years. So definitely a hot topic issue in the field, and really the sky's the limit. As you can tell, things in green were things that I found less than 5 publications, doing just a basic Scopus search or asking people at conferences recently. The main reason why you might want to do multimodal imaging is that there are fantastic temporal spatial trade-offs. So if we have here on the x-axis sort of the temporal scale, and on the y-axis we have our spatial scale, you can see that techniques like EEG and MEG give us fairly good temporal resolution down to sub-millisecond timing, if you can sample that quick. But fairly not great spatial information, although I'll make the argument that EEG and MEG have both gotten a lot better in spatial resolution recently. Whereas if we look at things like fMRI, we have very slow temporal resolution down to sort of multi-second integrations, because the hemodynamic response function takes about 8 to 12 seconds to rise to peak. But we get down to, say, layers of the cortex, and we can really disambiguate region A versus region B. What's also really fascinating about this chart is that this is the second publication of it in 2014. The original one in 1988, you can see we had many fewer imaging techniques, and the field looked considerably more sparse. Now we have a lot of different ways of sampling both temporal and spatial domains. And so we can do things with multimodal imaging that are sort of these information trade-offs. So we can use, say, EEG, ERP, and MEG to say when things happen in the brain. So at what point do you recognize a face? At about 170 milliseconds. At what point do you recognize that you've heard a sound that wasn't like the other sounds? About 300 milliseconds. We can also do things with fMRI, like where in the brain, where we can say, okay, now where in the brain do you recognize faces? Well, that turns out to be in the fusiform area, and some people have even dubbed that the fusiform face area. And then, of course, the neuromodulation, people are saying, well, what happens to a process when X area is disrupted? And of course, this is a pretty basic question. This is something that we struggled in neuroimaging for a while, is not so much what does region A or region B do, it's what does network A or network B do, and how do the regions within those networks contribute to that. So what happens to a process when you disrupt it, not just in space, but also in time? And there are some really interesting lines of research here where you can actually get people to, say, choose adjectives that describe themselves, and if you give them a TMS pulse at exactly the right time, you'll get people to flip their responses. So typically when you look at adjectives, you say, these are really complimentary. I want to be complimentary. I'm going to say, yes, this is like me. And if you zap them at just the right time, you can either slow down their sort of response to say, yes, I'm sure that I am friendly and outgoing, or you can even make them flip-flop and say, no, this actually describes my best friend more than it describes me. Also, don't TMS yourself at home. This turned out to be a terrible idea. So at the very surface level of all of this, pros and cons of different imaging, I feel like this table's been made 100 times, which is why you can see that I limited how many things that I put in. So again, EEG, high temporal resolution. It's also very comparable across the lifespan. So you can do EEG on infants and babies. You can do it on small children. You can do it on people who have metal in their bodies. It's also fairly inexpensive. So you can get started with a home-built Raspberry Pi style EEG system for about $100. That gives you about 3 channels if you want to experiment on yourself or your loved ones. But of course it has low spatial resolution, and there's arguments that source analysis is fairly difficult. MEG gives you also the high temporal resolution and arguably less difficulty in finding sources inside the brain because magnetic fields are not thought to be disrupted by the other parts of the brain, the skull, the scalp, CSF, and whatnot. There have been lots of arguments about this over the last 25 years. The downside is, of course, that MEG is expensive. It's about $2.5 million to buy your basic MEG system. It's difficult to use with children. You have limited signal from deep tissue or even surface tissue that just happens to have sources that don't cause the magnetic field to exit the head and then re-enter the head. That's how we measure everything. So if you have magnetic fields that just happen to be sort of tangential or oblique in different ways, then that may not get picked up by MEG. Hence, perhaps the argument for why you want to do EEG with MEG. fMRI, of course, has great spatial resolution, but it is also expensive, difficult to use with children, and gives us very low temporal resolution. And before someone objects and says, yeah, but we've got multiband and simultaneous stuff, remember that the blood flow measures that fMRI is really picking up on are limited by how fast they can move. So I've kind of set up this framework for why doing multimodal neuroimaging is hard, and we'll kind of step through them in different ways: design, data analysis, and interpretation. So in terms of designing multimodal neuroimaging, it starts off with this basic question, which is true of all science today, and something that we've talked about at some of the other talks this summer for the summer course, which is domain knowledge in a particular topic, and sort of having those theoretical questions and shaping them in such a way that they can be answered with neuroimaging, as opposed to, hey, I've got access to a $2 million or $8 million toy, and I'd really love to stick people in this toy and see what happens to their brains as we're having them do whatever, even if it isn't necessarily interesting or intriguing or extending science. You also have to have knowledge of some of the component parts. And so if you're doing, say, simultaneous MRI and EEG, or even sequential, non-simultaneous, you have to have some knowledge of how these technologies work, some knowledge of what they're good at. And I will take this moment to fully acknowledge that science is no longer, and arguably hasn't been done in a vacuum for a while. So it's no longer the case that you have solo researchers who are sort of going it alone. So these could be teams of people, but you still have to build up the teams that have the expertise to know these technologies. And of course, you have to have the technical knowledge to carry out some portion of the experiment. For anyone who's had their laptop die as they're about to present stimuli to a subject, you know the panic moment of what happens when technology is not our friend. Which leads to additional design questions like, do you have the right equipment to ask the questions that you're interested in? Do you have the expertise to operate that equipment? And is it worthwhile? So generally, when you ask me at a dinner party, is multimodal imaging always good? I generally say more data is always better. It is always better to have more data. Having more data that you don't know how to analyze, having more data that you don't know what's going to come from it is completely unhelpful. You could spend your entire career trying to fix by analysis that which is flawed by design from the get-go. So thinking out your questions beforehand so that you have a certain path to get there is always worthwhile. And then the final question is, can you do this well? And that is a interplay between the technology and you and your lab. So is the technology there to the point where you can ask these questions and you can really get at the information? Can you use, say, 7 Tesla MRI to get at layer-specific activations? Well, you can if you're the right person and you know how to do it. Can you pair that with EEG? You can if you have the right equipment. But the other questions to ask when we're designing multimodal imaging are a little bit harder, right? So they're both, what is the value added, and does it have to be simultaneous multimodal imaging? Because there's plenty of ways that you could do multimodal neuroimaging that aren't conducive to doing them simultaneously, or are much, much more expensive if you're going to do them simultaneously than sequentially. So for an example, if you were to do simultaneous EEG MRI, I would argue, and certainly the rest of the field agrees, that there's quite a bit of information that you can gather from having separate EEG and MRI sessions. But the real benefits of doing some of these things simultaneously is that you get people in the same environment, say doing EEG and MRI at the same time. So if you've ever been in a classic EEG experiment, you're usually sitting in like a lazy boy recliner in a comfortable room. It's quiet, it's dimly lit. The monitor is exactly some amount away from you. You can see it clearly. You're at no point wondering what that loud banging noise is. You go inside the scanner. I'm not a terribly large individual, but you're kind of snug. You feel a little bit swaddled by this $2 million magnet that's hugging you and aligning some percentage of your protons along an axis. You have those loud noises, and these are things that you want to be able to compare across the different techniques. The other things that we can do is, if you have simultaneous, you can look at how individual trials within a particular experiment change over time, and you can do it in both techniques. And that might be, say, activation, or sort of newer methods with coherence, resting states, inter-subject correlation, things like that. In terms of data analysis, this is where things also get hard, because you could have designed the perfect study and been told by your experts who comes to all of your lab meetings, oh yeah, we can totally do that. And then once you've collected all of your data, you find that maybe things are a little less cut and dry. So first of all, in terms of data analysis, if you're doing simultaneous EEG and MRI or simultaneous A and B, are there artifacts from this multimodal imaging method? And if there are artifacts, can you fix them? Because if you can't fix them, then you're just getting noisy data. So while you may be getting more data, which one would argue is better, noisy more data is probably not better. So as an example, in terms of artifacts in simultaneous EEG and MRI, we have 2 major sources of noise. The first is the MR artifacts caused by the gradients that make your EEG, which normally looks fairly smooth, look like this, sort of like an old TV when you didn't have good reception with the antenna. There are a few people in this room who remember those. Some of you might be too young. And the ballistiocardiogram, which is caused by, I will say, the fleshy parts of your face moving inside the scanner. Typically that happens when your heart beats. So every time your heart beats, the electrodes on your skin move just a little bit, and you get induction, which is the same thing that we use to power your sonic hair toothbrush, where we basically have a magnet and we're passing a wire through it, and so we get a blip in current. And that tends to follow at around 200-ish milliseconds, 250 milliseconds after the heartbeat. The good news is we're fairly good at removing gradient artifacts. So if we take the raw signal and we build what's called like a template of the MR gradient artifact, which is always the same every time, we can subtract that from all of the electrodes, and we go from something that looks really messy to something that looks fairly clean. The BCG is a little bit more difficult because it is not uniform over the entire head, most prominent on the facial electrodes, and while we can do really cool statistics like using a principal components analysis or independent component analysis to model the artifact, remove it, and reconstruct it, these methods are not perfect. And so pairing this analysis question with design, you may have to collect a few more trials. Somewhere around 10 seems to be the going rate right now, in order to get the same amplitude responses that you would because of this additional noise, even after we're cleaning out the artifacts. The next question is, if applicable, how will you combine this data? And the reason that I say if applicable is because with neuromodulation techniques, we could actually say that, well, we're zapping the person, technical term, at a particular time, and that's not something that we're necessarily recording. So do we really have to combine them a huge amount? Yes, but do we have to combine them as much as two recording techniques being done simultaneously? Not so much. So again, I'm going to continue to use EEG and fMRI as examples, but I could give you more examples of this if you want to chat after this or at any of the CMN functions. But how do you co-register data across modalities? How do you deal with, say, individual head models, individual subjects, or just taking everything at a group level and saying, you know, I took all my group effects in fMRI, I took all my group effects in MEG or in EEG, and they both kind of point to the same thing. I'll give an example of this later. And what are the trade-offs of doing sort of single subject versus group analysis? So again, in EEG, some of this is solved, and actually in TMS, some of this is solved too. So we can use different cameras and triangulate where all of the EEG sensors are on the head, and we can come up with a head model, and we can pair this with basically either a very generic shell model of the head, so we can say, okay, you have different rings of distortion, brain, CSF, skull, and scalp, or we can actually take your individual T1 anatomical, we can segment it into gray and white matter, and we can use that to figure out how much distortion you'd expect from different sources and how those sources might contribute to the scalp EEG at any given time. But then the difficulty here is, how do you really combine, say, EEG and fMRI? Because you'll notice here on the right, this is actually a movie of EEG going across the scalp in 256 electrodes, and this is the fMRI source analysis, or the fMRI analysis, and this is the EEG analysis, and you'll notice that, like, sure, I can get these maps to look fairly similar if I know roughly when to look. In this case, I knew that I was looking for a perihippocampal P300, which happens about here, and if I do my source analysis here, ta-da, I get an EEG source model that looks like my fMRI. But the trick is, I get an EEG source analysis model for every single time point. So if I'm sampling at 1 millisecond increments, then I have, say, 1,000 time points over 1 ERP that I could fit a source model to. How do you combine that with your fMRI? And historically, the way that people have done this is by taking the fMRI analysis, doing the EEG source analysis and saying, hey, look, we found the EEG that looks awfully like our fMRI. So we're going to say, declare victory, and ta-da, our fMRI must be correct, right? But the argument that I've made and several other people who will be at this October workshop, buy your tickets soon, they're free, there will be candy, is that the EEG itself is also picking up on additional signals throughout the head. And while we used to sort of shrug and say, yeah, these are probably noise because EEG source analysis isn't that good, what we're now starting to say is, well, actually, it could be that fMRI is missing a decent amount of sources that the EEG is picking up on. And so perhaps the question we should be asking is, when they diverge, what is interesting? And perhaps we should stop playing favorites in terms of which technology we like. My boss is not here, co-discoverer of fMRI, so I feel safe saying that. He'll listen to this later. But I'll tell him that everyone in this room was nodding. You guys should work on that. Yeah, good. Yeah. By the way, to those of you who are nodding constantly in the back of the room, you are the best people. Okay, in terms of interpretation, how do we interpret the findings? Well, I just gave you an example of simultaneous EEG fMRI where we get some of the same results and some of the different results. And do the analyses of your data let you make certain generalizations? So can you say, like, hey, we processed simultaneous data on a number of subjects, and we came up with roughly similar maps across them, and I'm going to use some figures to show this in a minute. But what does this really mean? Like, what sorts of nuances are we actually getting at with multimodal imaging? And honestly, I don't have answers for all of these. But as an example for interpretation, this is one of the seminal works, fantastic pieces of simultaneous EEG fMRI works in the literature all the way back to 2005, which whenever you think back to what year it is, I always think it's like 2005, 2006, and then I'm surprised when people tell me it's 2019. My checks get flagged a lot. So we have a flanker experiment. You have a series of arrows. Your entire task is to tell me if that arrow in the middle is pointing left or right, and obviously if the other arrows are pointing in a particular direction, whenever these are incongruent, your response time's a little bit slower, and you tend to make more errors, and you get feedback. And if we look at the EEG analyses, we see that we get a nice deviation fairly early on for the congruent versus incongruent trials. And we get a scalp topography that shows us sort of midline negativity, both to correct and incorrect trials. And if we use source analysis across our entire group of subjects, so we grand average all of our data together, we have one prototypical average, and we say, give me the source analysis of this, we end up with one source in the anterior cingulate showing nice conflict, which you would expect. If we take those same people who did this experiment in fMRI, and we do our standard deconvolution, we see that we also get an MRI source in the anterior cingulate cortex and some other stuff too. Notice how I downplayed that other stuff too. And this is how traditionally a lot of simultaneous work has been done, right? So it's, I have my results from something I'm really good at, and Bob down the hall had done the analyses from something he's really good at, and we looked at the maps together, and I kid you not, these things really happen. We taped all of the brain maps around the room, and we just let them sit there for 6 months, and then someone had the aha moment and said, that's what's going on. This is actually the story of the N400. Who, if you want more information, see me after class. But it turns out that, again, we're losing a lot of information because we're using the grand average, and we're coming up with a story based on glossing over all of the individual differences, and we're not really getting statistics that we can relate these two together because we have one number in EEG and we have one number in fMRI, right? Good job nodding, guys. So I think I've kind of outlined why multimodal is hard. And I don't think anyone's going to disagree with me that multimodal is important because it provides, hopefully, complementary and possibly overlapping information. It may facilitate better studies of individual differences because you can start to do things like use one method as a control within a subject, or as a modulator within a subject, or as another variable within a subject. And it can allow us to create for, let's say, multi-measure latent variable models, or if you've ever taken psychometrics, sort of measurement and variance models, where we can create latent variables based on some indicators from all of these different techniques, and whether we, say, do TMS at the right time or the wrong time, in the right place or the wrong place, and how that impacts the EEG and fMRI. And it may also be used to improve reliability and replication, which has the head of the data science and sharing team very alert right now. Good shout out. So again, these slides I've shamelessly borrowed from Adam Thomas on the data sharing and science team. And so if I have an fMRI study at P of 0.05 and I analyze all the data, and lo and behold, I have a picture of Peter Bandettini right here. We could call this Peter Bandettini on the brain, or the PAB area. And we might say, wow, this is a really crazy finding, and it's one subject, and then we could say run more subjects, and we might get different PAB areas, or we might get the same PAB areas across all of our subjects. But as Adam pointed out, it may be that other people run this study and they find that the Peter Bandettini area is in other parts of the brain, or it doesn't generalize in certain circumstances. And that's where I really think that simultaneous is going to be very useful, and certainly the publications are now coming out, both straight publications, but also computational model examples. Francisco's not here, but I'll poke him later, where we might be able to say, hey, in participants who have a fMRI Peter Bandettini area in this area, their EEG looks like this over certain electrodes. Or say we get source analysis that gives us a Peter Bandettini source analysis with a particular frequency. Sorry, I didn't draw a face in the EEG. That's on my list of to do for next year. And it could be that we follow this up with an MEG study, and again, with the same subjects, we find that there are particular signatures that are unique to individuals that show one activation area versus another activation area. And so now we start to get multiple measures that are going together. And I should have made this fade in earlier, or later. What is the probability of finding overlapping significance? Well, if these were all independent measures, like a coin flip, you would just multiply those together and you would come up with a particular number. But here you have a dependency structure that is honestly a little bit hard to model, although I was told earlier today by Gong Chen that apparently it's no longer a big deal. And he gave me this hint that they're not fully dependent, and they're also not fully independent measures. And so the probability of, say, finding any two of these might be somewhere around 0.005 instead of 0.025 if you multiplied them together. So as I said, I don't have all the answers, fortunately, otherwise I'd be very bored in my job. And, well, someone should do other things. But just some thought questions for you. In terms of design, we have yet to really answer this, what questions best lend themselves to multimodal imaging? Because you could just say, welcome to NIH. We have simultaneous EEG, we have simultaneous TMS and fMRI, we have simultaneous MEG, TDCS, TACS. Go to town, collect all the data that you want. But are there particular studies that really lend themselves to this question of, can we use different methods to get at the same or slightly nuanced questions in ways that are better than other studies? There will be an entire panel on this at the Multimodal Conference. Next one is, how should analyses be standardized to make better use of multimodal imaging? And one of my all-time favorite things about science is this advent of open source software, and the fact that you can go on the web and you can download MATLAB or Python toolboxes that do data analysis, and they've all implemented what they think is the best, air quotes, way of doing a particular thing, and oftentimes these are non-overlapping best ideas or best practices. And the question is, should we standardize them? And this, I think, is going to lead to a vast array of research in terms of comparing certain models. So in EEG, we can do source analysis using either that sun shell model that's very obtuse, very like here are 4 or 5 shells that model different resistances. We can do finite element models where we only project the surfaces, or we could do finite differences models where we can project the volume, and that projection changes from millisecond to millisecond depending on what sources inside the brain are active at a given time. And these are definitely open questions. How should we standardize? And how are we going to declare sort of a winner, if there is such a thing as finding winners in science? And the other question is, who's going to do this work? And I'm looking at every person in this room. Not it. Kudos. Other people who touch their nose just then. And then the final one is interpretation. So what will be the scientific or statistical level of rigor to support any kind of multimodal imaging conclusions? So at what point do we stop cutting people slack in multimodal imaging for saying, hey, congratulations, you put 2 imaging things together? Todd Constable, the imaging director at Yale, once told me that back in the 90s, you could publish your grocery list with a brain figure next to it in almost any journal because it had brain pictures and people were really excited. He might have been a little facetious about this, but nonetheless, it kind of rang true. And whenever you went to conferences and you saw brain pictures on posters, you went towards the brain pictures and you left poor old me by my EEG poster all alone and sad. And I'm holding that against the field right now. Maybe just a little bit. Well, rewrite Dear Evan Hansen, all about EEG. But we do need to come up with a method of really measuring what is statistically significant and what is a measurable advance. And there are already arguments in the field about doing away with p-values. Highly recommend the book, What If There Were No Significance Effects? And most of those use effect sizes, and then we could get into the arguments of big effect sizes versus small effect sizes, and what you can reasonably do with, say, n equals 20, 40, 60 subjects, which are sort of the norm in science, versus n of 600 or n of several thousand, which are becoming these sort of UK Biobank or HCP or ABCD, ADNI, things like that. And these are all kind of fundamental questions of where we go forward. So if you have any of the answers to these, you can slip it anonymously under my door. That'd be cool. Or send an email or come do a study with us. We at the Center for Multimodal Imaging are always eager to help you do multimodal imaging. Literally, we'll teach you how to do most of it, teach your ERDAs and postdocs how to do it. You'll get your fair share of troubleshooting when things break, but it'll make you a better person overall. It's like eating vegetables. And with that, I'll take any questions. Bueller. I like when people smile to that. It tells me that you're still watching that movie. Yeah.
So in an ideal world where we can do multimodal imaging and it is something that is standardized and where we know that it is good, what practical uses do you see more? Do you see it being used like pathology? Do you see it being used with diagnosis? Do you see it being used more for just learning about the brain?
I will pay you for this question after, as I promised. So yes, multimodal imaging will likely lead towards clinical applications. The very clean, very prominent signal out in the field is that while fMRI has not found a good clinical market, many of the other techniques in the field have found good clinical uses. EEG has been shown to do, say, identification of seizures, has been used to diagnose whether or not you will have a reading disability. When we test you at birth, we can tell you with about 90% accuracy if you will grow up and have a difficult time reading. EEG has been shown to identify Parkinson's before you might otherwise pick up on it. MEG has been used to localize language. So if you ever go in for, say, a brain surgery and they're worried about cutting out your language area, they do this thing where they do a WADA test. So they take some chemical, they inject it into your carotid artery on each side, half of your brain goes to sleep, and if you retain the ability to speak, then they say, aha, language is on the other side, or the side that's not anesthetized at this particular moment. The downside to the WADA technique is that not a non-significant number of people who undergo this technique have a mini stroke or other adverse reaction to it. Medicine is incredibly slow to change, which is why MEG has not been adopted more in this, and fMRI has also been shown to be fairly good at this as well, but MEG and EEG are certainly kind of leading the way. But I believe that if you are going to find a good clinical case for fMRI, you're probably going to find it as a value added first in terms of other techniques. Now, I know this is a race, and like someone in my lab will probably beat me to this, using fMRI to do individual differences in people with patient populations, but nonetheless, I think that one of the easier ways to get there is to say, okay, we know these markers exist in EEG. What do we see in fMRI? How do we get that value added from fMRI? Or how do we get that value added by disrupting the signal with TMS or things like that? So thanks for that question. Anyone else? Okay, guys. Thanks so much for coming.